Hello, in this video, we will talk about a journal article. Whole genome sequencing of a Plasmodium vivax clinical isolate exhibit geographical characteristic and high genetically variation in China-Myanmar border area by Shen Pochan Yue Wang, Kokui Kasene, Ping Xu, Hai Mo Shen, and Junhu Chen. This video is solely for seminar purpose and we don't claim any credits for the finding in the journal. Our team members will be Mi Chan, Shahjahan, Siwaraman, and Praveen. The introduction of the journal informs us about the background of Plasmodium vivax. Plasmodium vivax being the widespread malaria species, by studying the parasite, the researchers found that members of the multi-gene families are genetically variable. The genetic variability may be the cause of the drug and human immune response evasion. The Plasmodium vivax is not amenable in vitro culture with leukocyte depletion. Then the journal talk about sequencing and notation of first Plasmodium vivax obtained from China-Myanmar border, CMB1. The map and de novo assembly shown a similar result confirmed in the past with high sensitivity detection. And they also search for genetic codes that are unique in few individual, less expensively, and higher effectiveness. The methodology used by the team was genomic data, collecting and sequencing the sample, identifying single nucleotide polymorphism, and to confirm order of sequence of the DNA, de novo assembly and gene predictions are used. The genomic data used were obtained from the GenBank and Plasmodata Bank, published from monkey-adapted strain, the Plasmodium vivax, and also referred from 6 human isolates. These are the simple sequence repeats referred from GenBank. Sample collection are done by extracting the blood of malaria-infected patient. The sample is confirmed as Plasmodium vivax monospecies infection by PCR-based diagnosis, and the DNA was extracted using Jiachan DNEZ blood and tissue kit.
Then sheared the genome into 500 base pair fragments using a Covarice S2 instrument to construct the Illumina sequencing library. It will insert size into 250 base pair. Adapted sequence and low quality sequence removed using 3-momatic 3.0.
In identification of SNP and indel, their map sequencing route from all samples to the Plasmodium vivax Salvator 1 genome using PWA and SAMTL 1.3. In their study, SNP was defined as nucleotide position covered at least 10 reads in at least half of analyzed sample. To identify insertion and deletion, which is indel, we have followed the procedure described by Chan et al. Briefly, we analyze all read pair that do not map in the expected configurations and might be indicative of deletion and inversions.
In identify SNP from China et al. Nucleotide position covered by at least 20 reads with a quality score greater than 30 focus. Consider only read pair that map in head-to-head configuration. Characterize the function of each DNA polymorphism identified using the Salvador 1 gene annotation downloaded from Assemble. In de novo assembly and gene precision, host DNA sequence removed by aligning all clean pair and reads to Homo sapien genome using top hat 2.0, and the non-aligned reads were considered to Plasmodium vivax reads. It is to determine the optimal k-means, different k-values tested, and compare the resulting assemblies. Sequence of the whole virgin family were collected and compared using by BLAST and MEME to find all potentially virgins. An ab initio gene prediction process was performed using the Augustus and Gilmer HMM for all contigs. The remaining genes were finally compared with the Swiss prot and an R database by BLAST. All partial genes without varied BLAST result discarded and consider the rest as novel gene. The results show the summary of sequence map. The average genome coverage was 62 times, although it was variable in subtelomeric region.
The table shows sequencing and mapping summary statistics for samples from field isolate and monkey-adapted strain. Telomeric deletion in Plasmodium vivax strain. South America strain has very few hits that could map to a 130 kilobase region at the subtelomeric end of chromosome 7 due to a sharp decline of GC content along this subtelomeric region and accompanying enrichment of repeated sequence. CMB1 isolate maintain genetically homogeneous. Meanwhile, the subtelomeric deletion occur in North Korea and Cambodia isolates.
High genetic diversity of Plasmodium vivax CMB1. From 108,846 nucleotide variants, an average of 4.84 SNPs per kilobase identified. 33,000 of these SNPs are positioned in the coding region of 3,986 genes on 14 chromosomes. There were about 27,000 non-synonymous mutation and 6,000 synonymous mutation. And from these indels, they were in the coding region of 1,707 genes. These highly polymorphic genes are associated with red blood cell invasion and immune evasion as shown in the next table. Genetic variation gene and SNPs. From the table shown, about from 5,626 gene, there are 5.97 SNPs per gene in average. And it's worth to note that reticulite binding protein and merozoite surface protein 3 shown a rather high SNP per gene, about 70 and 55 each. Plasmodium vivax distinguished by geolocation. Spearman correlation matrix explain a high ratio which separates the Americas from Asia. PCA results show that P vivax clustered generally according to the geographic origin. The phylogen tree was clearly clustered into three groups, including Asia, Africa, and South America clades. India's strain was tagged under Africa category instead of Asia and more close to South America clades in PCA approach. PCA analysis of Spearman's correlation matrix. From principal component analysis based on 100,000 SNPs in field isolate and monkey-adapted strains, the colors correspond to the geographic origin. Blue for Central and South America, green for Asia, and gray for Africa, and black for India. By using the maximum likelihood technique, the neighbor joining pylograms indicates the geographic origin based on lineage, and these are constructed from the 100,000 SNPs. The branch lengths indicate the considerable diversity of the PY strain, and it's worth to note that India strain was tagged under Africa, as mentioned before. Standardized genetic marker identifying genetic signature. The existence of 42 SN barcode as marker identified genetic signatures. The SNPs were divided into three geographical groups, and 539 of them showed consistency of geographic distribution. SNP markers are very effective in distinguishing the African and Asian sample, but not yet support the New World sample signature. Single nucleotide polymorphism as baseline to distinguish geographic origins in Plasmodium vivax. As shown in the table above, you can clearly see the SNP allele shown in the India is quite different from the SNP allele shown in North Korea, distinguishably colored by the green and yellow colored marker. De novo genome assembly of Plasmodium vivax CMB1 composed of 8,400 contigs with average GC content of 39%, and 29 billion of the estimated length assembled with 5,700 contigs, and 5,966 contigs are mapped to 14 chromosomes of Salvador 1 genome using BLAST+. They account for 99.8% of the 14 chromosome, an overall length of 95%. And the ab initio gene prediction was the method used to predict the partial gene loci. And they identified 661 novel PVAX gene, with 32 similar to established and 258 similar to hypothetical proteins.
The virus superfamily is distributed in a variety of terms and codes proteins which are exposed to the host cell to avoid an adaptive immune reaction by host. Plasmodium vivax cell 1 is classified into 12 subfamilies with 346 virus genes. This figure shows the motifs of novel Ver gene in subfamily J. The horizontal line depicts the number of domains, estimated location in the gene sequence, and the different colored squares shows the corresponding motif in part A.
One of the main purposes of this study was to identify whether the simplified sequencing approach caused systematic errors in genomic level. Sequencing the Plasmodium vivax genome has provided insights into parasite biology, but have also caused many challenges as listed. Myanmar was considered as the main imported source of vivax malaria, and sequencing analyses have identified greater levels of genetic variability. Myanmar was considered as the main imported source of vivax malaria, and sequencing analyses have identified greater levels of genetic variability. In this study, they have presented a 539 SNP dataset for Plasmodium vivax that spanned all 14 chromosomes of the genome and involved 267 genes, with the intention to increase sensitivity and detect genotypes with higher accuracy. In a nutshell, it can be concluded that the direct sequencing approach used could produce more available reads for mutation detection and be used as an essential tool in near future to provide important genomic informations of vivax malaria. These are the remarks and comments pertaining to the approaches and methods used. The shotgun approach with clone counting method using Illumina semiconductor sequencer and DNA chip for SNP analysis to identify the mutation and serve as DNA markers. That sums up our entire presentation. Thank you and stay safe.